Hey guys, welcome to Human Physiology Bio22. This is Kate, and I'm recording this lecture on Wednesday, January 29th, specifically for the spring 2020 semester. So over the past couple of weeks, we've been talking about digestive anatomy and physiology and getting into metabolism. In lecture specifically this week, we talked a little bit about protein metabolism. So once we get this big macronutrient, we break it down into smaller pieces and absorb it into our body. How does our body work with those little building blocks? So we talked about protein. And in lab, we've talked a little bit about carbohydrates, specifically in terms of metabolic states. So we're going to expand on that a lot today and talk about carbohydrate and lipid metabolism, specifically the relationship with acetyl-COA and working up to a diabetic ketoacidosis case study. So we're continuing kind of these week 3 learning objectives, especially getting into these key pathways or destinations or outcomes for those building blocks, those monomers, the byproducts of macromolecule digestion. So this really relates to a lot of those specific learning objectives and expands on them. So remember, we're tracing those key pathways, those outcomes. We're practicing identifying anabolic versus catabolic reactions based on whether energy is used or released. We're thinking about what happens when those key carbohydrate metabolic pathways are in action and kind of what they're doing, and whether specific pathways are more likely to occur in absorptive or post-absorptive states. So here it's important to remember from lab that when we say post-absorptive, we can mean fasting or we can mean starvation. So those are both technically post-absorptive, but they're a little bit different. So we have to distinguish what pathways are happening at any given time. So we're thinking about glycolysis, gluconeogenesis, glycogenesis, and glycogenolysis. And remember, we practice breaking apart those words so that we're able to really think about what that word is telling us in terms of the process. At the end of the lecture, I'll introduce this diabetic ketoacidosis case study. So you should think about the role of acetyl-COA in that whole process, how it relates to the Krebs cycle and to aerobic cellular respiration. We need to be able to relate acetyl-CoA to lipid metabolism. Oftentimes we think about acetyl-CoA, it gets grouped into the kind of these reactions that start out with glucose. And so I think a lot of people think about carbohydrates and glucose for energy, but maybe don't acknowledge that acetyl-CoA really ties carbohydrates and lipids together, and that a lot of molecules, even though they're chemically very distinct, can go through a series of reactions to, become one another and cycle into these pathways. So we'll relate acetyl-CoA specifically to lipid metabolism and think about, the physiology of this, specifically disease pathology. When you have a diabetic individual whose homeostasis is thrown off, how does that play out? Remember, when we absorb nutrients into our body, these broken down monomers, when we get them into our body, they get cycled into circulation, either in the circulatory system or the lymphatic system. And once they've been detoxified and processed and enter fully into circulation, there's a few different ways our body can interact with those monomers. So we can use them for energy. We can cycle in carbohydrates, proteins, and fats, those pieces, into energy producing cycles. We can also use vitamins and minerals that we absorb as enzymatic cofactors. So remember, a cofactor is an inorganic substance that kind of helps the enzyme achieve the correct shape. When we say a coenzyme, we are usually talking about vitamins, different organic molecules. So I did want to just briefly clarify that. Sometimes cofactor and coenzyme get used interchangeably. There is a slight distinction, but they both speak to this idea that when we eat food, when we absorb material, we're not just doing that for the macromolecules. Those trace elements, those molecules or micronutrients are also very important. We can also use what we absorb into our body to make up components of our cells or our interstitial fluid or important functional molecules. So we have these biosynthesis reactions, those anabolic processes that may end up being very structural or functional. And then we also can store it. So we'll talk about specific storage forms of carbohydrates and lipids. So this is, again, an anabolic process. We're taking those small pieces that we've been able to absorb, packing them together to have these storage forms of fat or glycogen, which is a complex polysaccharide. So we're kind of hanging on to that stuff. We have extra stuff. We don't want it to be kind of removed through our excretory or excretion systems. We want to hang on to it so we can use it in the future. So we're going to focus first on carbohydrate metabolism. This is something that we did cover quite a bit in lab, so it's kind of a review, but kind of going a little bit more in depth. Specifically in terms of glucose, which is, remember, a monosaccharide, a single sugar ring, a simple sugar, and glycogen, most organisms have ways of packing glucose together, whether it's amylase, amylopectin, so different types of starches, or cellulose, or chitin, or glycogen. So animals use glycogen as a storage form of glucose. It's how we pack things together. So remember from lab, we talked about these 3 different metabolic states. We can broadly distinguish it into whether it's absorptive or post-absorptive. For post-absorptive, we're thinking about whether it's fasting or starvation. So there's kind of different levels of that post-absorptive state. Absorptive can also be called fed state. It's when you've absorbed a bunch of material and your anabolic processes are exceeding your catabolic processes. So that's one thing I want to point out, is that in general, in absorptive states, you have more of this anabolism happening, and in post-absorptive states, you have more catabolism happening. You have to access those stores because you don't have those dietary sources coming in. That being said, in absorptive states, you always have catabolism happening too. It's just that anabolic processes are kind of outweighing it. And in post-absorptive states, you also always have anabolic processes happening. It's just kind of looking at this big picture and thinking, what's the relative ratio? What's really outweighing or dominating in the situation? So when you're in an absorptive or fed state, you have a lot of these extra building blocks. You can pack them together. In general, more anabolism is happening than catabolism. But many hours after you've eaten and you've packed away some of these reserves, it starts switching so that catabolism is exceeding anabolism when you get into those fasting states. So you're starting to have to access and tap into those reserves. Then when you get into starvation, that's really survival mode. Your goal is to just make sure you have enough glucose to power your brain. That's like the bare minimum, and you have to start using really unique catabolic pathways that we don't often think about. So there are some differences in what's going on between post-absorptive fasting and post-absorptive starvation. So remember, when we think about absorptive state, and I know that this image is kind of hard to see, these figures are displayed a little bit bigger in the in-person lecture from this week, and they're also available in your textbook for chapter 23, pretty large. So in general, it's just kind of thinking, you know, like in your liver, in your muscle, in your adipose, where you have these reserves of glycogen and of fat. How are they changing? Is our body kind of using those reserves, packing away or breaking apart in absorptive versus post-absorptive? So when you think about absorptive, and we're focusing here, remember, on glucose and glycogen, absorptive, you have a lot of blood glucose. So you have a good amount increase in blood glucose. That means that's something that your body can easily sense. When you have the circulatory system, a lot of organs can kind of monitor that and pay attention. They know, like, okay, you have plenty of blood glucose, so we can start packing it away. So remember, you don't want to just keep packing it away. There's those negative feedback loops involved. It's a delicate balance. In post-absorptive, you don't have enough blood glucose, so then you have to start bringing it back up. So again, it's that seesaw of homeostasis. So these kind of reactions that are happening, these catabolic reactions that are happening in post-absorptive states, are to tap into those reserves of glycogen in your liver and muscle, and then fat in your adipose cells. So remember, glucose is a monosaccharide. It's a very simple ring structure. It's a sugar, and it's stored as glycogen, a polysaccharide, in your liver and your skeletal muscles. So I think a lot of us think about it as being in our liver. A big fraction of it is actually in our skeletal muscles. It's involved in that fight or flight response. But glycogen and like kind of these glycogenic and glycogen-related processes really do focus in the liver. There's also interaction with the pancreas. In the in-person lecture, we talked about glucagon and insulin. Glucagon is a catabolic process. It stimulates that breakdown of glycogen. Insulin is an anabolic process. So when you have high blood sugar, insulin is important in packing away that blood sugar into glycogen. If you have low blood sugar, glucagon is important to break apart that glycogen and increase your blood sugar again. So remember, there's a few different G words that all sound pretty similar: glycolysis, gluconeogenesis, glycogenolysis. So I think a lot of the times when we're studying, we look at a word, we're like, okay, I got the first GL part and the rest kind of blurs together, right? And so that's why when you're trying to memorize things, it can get really dicey if you have all these words that look very similar. So remember, I really want you to take a moment, pause, and start cutting apart that word. With glycolysis, we can split it into glyco and lysis. Glyco is telling you sugar, glucose. Lysis is telling you splitting. So here we have the breakdown of glucose to pyruvate and ATP. Gluconeogenesis, gluco is again glucose, sugar. Neo is new, so like neonatal or neo-Nazi, it's a new version of something. So neo is new. And then genesis is creation. So this is the new creation of sugars. It's de novo, completely not from dietary sources. It's other different substrates that you have already in your body being transformed in different ways to result in glucose. Glycogenesis is glycogen genesis. So it's the creation of this sugar storage molecule of glycogen. So you're forming glycogen, a very large energy storage molecule, from these simpler sugars from glucose when, so that should actually say when blood glucose is adequate. So I will re-upload these slides and I'll fix it in a few different places. Sorry about that. So when blood glucose is adequate, when there's a lot of it, then you can start packing it away as glycogen. When blood glucose is inadequate is when you have glycogenolysis. So that's glycogen lysis. So it's the storage. So glycogen is the storage molecule. Lysis is splitting. So here you're splitting that glycogen to increase blood glucose when it's inadequate. Sorry, even if you've been teaching this for a very long time and you know these words, there's a lot of things that sound similar and it's easy to have a mistype. So I apologize for that. Glycogenesis is when you have plenty of blood glucose, you can pack it away. Okay, and then for, this is also from the lab, so this is kind of that answer key. So I typed it correctly here, glycogenesis, when blood glucose is adequate. So sorry about that. But what I wanted to point out here is that when you're kind of labeling something as catabolism or anabolism, it's very challenging to just say catabolism is always happening in a post-absorptive state, anabolism is always happening in an absorptive state, because there is some overlap. So for example, glycolysis happens in the absorptive state, and in post-absorptive fasting, it shuts off in post-absorptive starvation. Gluconeogenesis is anabolic. It is happening actually in a post-absorptive state. So even though we think about post-absorptive as being more catabolic, here you are making these completely new sugars, and it's technically an anabolic process, even though it's happening in the post-absorptive state. Glycogenesis is anabolic, that is absorptive. Glycogenolysis is catabolic, that is post-absorptive. So that direct relationship between anabolism and absorptive and catabolism and post-absorptive is very clear in the context of glycogen. It's a little bit trickier when you're thinking about glycolysis and gluconeogenesis. Okay, so I'm going to focus first on cellular respiration. This is going to be a review for you from Bio 5 and anatomy and all kinds of other classes you've probably taken already. For me, the goal of learning about cellular respiration is to think about what processes your body is relying on, how it's kind of transitioning between starting and ending molecules and what it's generating, and also thinking about coenzymes like NAD, NADP, and FAD. So here I'm thinking about kind of bigger processes. This is not a biochemistry class or an organic chemistry class. I'm not expecting you to know every single step of these processes, just kind of the big picture ideas. So when we're thinking about cellular respiration is just this generation of cell energy, and it can mean a lot of different things. So when you have kind of like anaerobic respiration, you're thinking about fermentation and all kinds of other pathways. Generally, when we're thinking about cellular respiration, we're usually talking about aerobic respiration. Remember, glycolysis does not rely on oxygen, so that is something that can run regardless of whether you have oxygen in your body or not, but it does shut off during starvation. So cellular respiration starts out with glycolysis. We have the citric acid cycle, which has a bunch of synonyms. It's also called the Krebs cycle, the TCA cycle, and that starts with acetyl-COA. And then we also have oxidative phosphorylation, which involves electron transport and chemoosmosis. That is where you get the bulk of ATP being generated. The citric acid cycle is important for really getting a lot of those high energy molecules, those carrier molecules. And then this figure is actually kind of lacking something. It's missing the relationship between glycolysis and the citric acid cycle, which is the transition reaction. So to actually go from glycolysis to the citric acid cycle, you need this processing of pyruvate, so this transition reaction. So we're actually going to call glycolysis stage 1, the transition reaction stage 2, citric acid cycle stage 3, and oxidative phosphorylation stage 4. So when we're thinking about glycolysis, it's going from glucose to pyruvate, and you end up producing a net of 2 ATP and 2 NADH. So here it's like you're charging your battery, you're filling up your wallet, right? So you're getting some of that stored energy. You are taking 1 glucose and ending up with 2 pyruvate molecules, which is useful and can be processed further, but again, something has to happen to the pyruvate first. Glycolysis doesn't take place in the mitochondria, it takes place in the cytoplasm, and remember, you don't need oxygen to do this, so it's anaerobic. It's going to be happening regardless of whether you have oxygen in your cells or not. It's just a matter of whether you have glucose as the original starting substrate. So when we're thinking about a lot of these cellular respiration processes, we're thinking about the sugar source, the chemical energy source, as well as oxygen. So I mentioned that stage 2, that pyruvate oxidation, is really important to transition between glycolysis and the citric acid cycle, or the Krebs cycle, or the TCA cycle. On an exam, I will write out all those synonyms so you remember that those are all the same things. It's really challenging to really assess your understanding if I only say Krebs and you forget that that's the same thing as the TCA or the citric acid cycle. So I'll try to anticipate that and account for that. So between glycolysis and between the Krebs cycle, we have this pyruvate oxidation, this transition reaction occurring. We're starting out with pyruvate, we're ending up with acetyl coenzyme A. So the reason for that, the reason for making acetyl-CoA and modifying or changing that pyruvate is acetyl-CoA is the first reactant in the citric acid cycle. So you can't start that next step. You can't go beyond this very small production of ATP in glycolysis if you don't transform the products of glycolysis first. So that acetyl group that is in pyruvate has to be activated, it has to be oxidized in order for us to be able to proceed with the Krebs cycle. So that carboxyl group is removed from pyruvate, that releases carbon dioxide. Remember, a lot of these processes release carbon dioxide. So we have that NAD becoming NADH, so we're generating a high energy carrier molecule, and we're transferring an acetyl group to coenzyme A. We're getting ACETYL-COA. When we're thinking about this process, there's a few kind of things to consider. We have this crossing from the cytosol into the mitochondrial matrix. So I'm hoping that this is going to repeat after it goes through the whole Krebs cycle. Maybe not. Yeah, it's sometimes hard to use GIFs in here. But anyway, so you can kind of see that pyruvate being changed and the acetyl group being added to the coenzyme A, and then kind of cycling in, interacting with oxaloacetate and starting this cycle. Okay, so then as we get into the Krebs cycle, the citric acid cycle, that's happening in the matrix of the mitochondria. So we have that acetyl-CoA, it's being changed in different ways. We have oxaloacetate, that carbon group, and that's going to be regenerated at the end of the cycle. So we have them coming together, and we have a lot of NADH being produced. So let me kind of indicate that here. So we have NADH over here, we have NADH over here, and over here. We have FADH2. So remember, that's a lot of charged batteries. It's a lot of electrons and hydrogens that are then ready to be cycled into future reactions. We also have carbon dioxide over here and over here. So we have some of those classical waste products of cellular respiration as well. So when we have this process, we're taking water and acetyl-CoA, and we're ending up with carbon dioxide as, like following the carbon molecule or the carbon atoms, but we're getting an okay amount of ATP and a lot of NADH and FADH2. So the Krebs cycle, it's kind of like glycolysis is kind of like if you're on a roller coaster, it's like that first kind of small dip, and then going up the Krebs cycle, it's kind of like, that's like glycolysis, and then now here we're like really ramping up. We're getting a lot of that stored energy that we're going to be able to then use to generate a ton of ATP once we get into oxidative phosphorylation. So remember, things like NAD, NADP, and FAD are all coenzymes, and they're able to take on hydrogens and electrons and move them around. So they start out NAD, NADP, FAD as like a dead battery or an empty wallet, and then they're able to take on this hydrogen, take on this electron, and become a charged battery that can then be used, or a full wallet that can then be spent. That energy is being stored in bonds of ATP, NADH, NADPH, FADH2, and then they're broken apart. They release that cellular energy. Okay, so then finally getting into stage 4, the electron transport chain. This is happening in the mitochondria, mostly in the matrix and the intermembrane space. So remember, mitochondria have a double membrane. So they have this intermembrane space. So this goes cytoplasm, outer membrane, intermembrane space, inner membrane, and then the matrix on the inside. So we have all of these proteins embedded in that inner membrane. And what's happening is as we have the electron kind of cycling through that electron transport chain, the hydrogens, when you pop off the hydrogen and free the electron, the hydrogen, the proton, is going into that intermembrane space. So you end up over here, I need to get a better pen to draw in here. You end up with a very high concentration of protons in that intermembrane space, a very strong concentration gradient. And remember, strong concentration gradients represent the potential for change. So you have this accumulation, this stored potential. The electrons are pulled through, and then oxygen receives them. It's the terminal electron acceptor that produces water. So that's why water is a waste product of cellular respiration. And then all of those hydrogens are going to flow through ATP synthase or ATP synthetase, which is then going to make a bunch of ATP. So that's an enzyme that's embedded in that inner membrane, and it's really a bunch of ATP. It's 32 to 34 net ATP. Just to kind of visualize that, on this left image, we have those H+, those protons going, as we go through these different complexes in the electron transport chain, those are shuttled into the intermembrane space and really build up that, that proton gradient. And remember, all of those FADH2 and NADH are coming from the citric acid cycle. So the citric acid cycle builds up that stored energy that's then popped off, and then the electrons cycle through the electron transport chain. The hydrogens go into that intramembrane space. So those hydrogens, those protons, then flow through ATP synthase, which is that enzyme embedded in the membrane, and that's going to power this production of a high energy bond between ADP and inorganic phosphate. So that's what we see here in this image. We have ATP synthase. The protons are flowing through. That flowing through allows this enzyme to create a high energy bond between ADP and inorganic phosphate, and then we get ATP. So I wanted to kind of help visualize that a little bit more, how it actually looks, because the enzyme structure is very unique. And this idea, this specific part of oxidative phosphorylation is called chemoosmosis. So we kind of use electron transport chain, oxidative phosphorylation, chemoosmosis interchangeably, but really the overarching thing here is oxidative phosphorylation. The two components to it are the electron transport chain, which is speaking to the electrons, and the chemoosmosis, which is speaking to the protons. Okay, so this is just kind of an overarching summary of glycolysis, pyruvate oxidation, also called the transition reaction, citric acid cycle, also called Krebs or TCA, and oxidative phosphorylation. So it's also important to remember that around 40% of the energy that is stored in glucose is captured as ATP, but the rest is actually lost as heat. So we talk about this transformation of energy and matter, but it's not a perfect process. A lot of energy is lost. Okay, so getting into processes of lipid metabolism, so thinking about acetyl-CoA, going back to that transition reaction, remember that acetyl-CoA is produced from those, when you have glucose originally from carbohydrate metabolism, that glucose is broken apart in glycolysis, you end up with pyruvate that is further oxidized to acetyl-CoA. So we know there's a direct relationship between acetyl-CoA and carbohydrates, but there's also a relationship there between acetyl-CoA and lipids. So acetyl-CoA is actually called a central intermediate. It's kind of this in-between stage of a lot of steps of lipid metabolism. So when you're going between fatty acids and ketone bodies, we have acetyl-CoA. So this figure has a lot going on in it. I'm going to explain some of these terms and kind of relate them all back together at the end of this portion of the lecture. So lipid metabolism is involves kind of this processing of fatty acids. So remember, when we have lipids or fats, they consist often of triglycerides. Those triglycerides are then further broken down into monoglycerides and two free fatty acids. So we're thinking as part of the whole metabolism, how we're getting back and forth between bigger molecules, those kind of like ketone bodies or full lipids, and fatty acids. So when we're thinking about lipolysis or lipolysis, again, we need to break that apart. It's lipo and lysis, so that's breaking down of fats. So this is taking triglycerides, taking that monomer that is making up the lipids, and then further breaking it down. So once we've absorbed that component into our body, how do we kind of hydrolytically break it down further. Remember, enzymatic hydrolysis is breaking things apart using water. So in this process, we have those triglycerides, we break them apart further into glycerol and fatty acids in the cytoplasm. So you then after you have those different components, you can take it one step further and use something called beta oxidation, which is then going to produce a lot of acetyl-CoA. So ultimately, when you have these different processes, when you have lipolysis, when you have beta oxidation, you're going from the components of lipids to a bunch of acetyl-CoA, and that helps prime you for the Krebs cycle. So remember, even though we think a lot about carbohydrates as feeding the Krebs cycle, other components can also feed into it. So conversely, when you're taking these small pieces and packaging them together for storage, that's lipogenesis. Remember, lipo is fat, genesis is creation, so this is storing things as fat. We have our fat reservoir over here, you can see. So we have adipocytes, those storage cells. So this is an anabolic process. It's building something bigger. Excess acetyl-CoA is converted into fats, so things like fatty acids, triglycerides, cholesterol, sterols, and bile salts. So here again, we're kind of tying together all these unique fats. Remember, lipids are broadly these hydrocarbon structures that are hydrophobic. So we group ring structures like cholesterol and things that are made up of triglycerides, other types of lipids together, but there are differences between them. But again, they can all be formed from acetyl-CoA. So that's happening in the cytoplasm of adipocytes, so fat cells, and hepatocytes, liver cells. I also wanted to take a moment to define this term ketone body. It sounds very fancy, but we can break it apart. So ketones are functional groups. You may or may not have learned about this in your chemistry classes or in biochemistry, but basically, remember when we draw chemical structures, since a lot of things are hydrocarbons, we don't write out the C's and the H's. We're just kind of implying them when two lines come together. So here we have a carbon that is double bonded to an oxygen, and then there's two additional R groups. Carbon is able to form 4 bonds. We have 2 of them going to oxygen, 1 to the first R group, another to the other R group. So these residual groups, these kind of unique changeable structures. So that's the basis of a ketone. When we have ketone bodies, these are just bodies. They're little structures that are ketone-based molecules. They're produced by the liver from fatty acids. So when you have different fatty acids, the liver can go through this process of generating ketone bodies and then having these like molecules with ketones as the kind of basic structure. So one example of that is acetoacetate. It has, a ketone, well, it has a couple ketones actually. There's beta-hydroxybutyric acid, which has a ketone, and then another kind of similar looking structure, but with a single bond instead of a double bond. And then acetone, that's a common one that a lot of you have probably heard of. So these ketone bodies are kind of a accumulated form of this like breakdown and metabolism of fat, but they can then be turned into acetyl-CoA. So you can produce ketone bodies from acetyl-CoA, and then you can also convert ketone bodies back to acetyl-CoA during post-absorptive states and stressful situations where you need a quick access to acetyl-CoA to power the Krebs cycle. So lipid metabolism also involves kind of this oxidation of fatty acids, and continues this idea of working with ketones. So when you have that excess acetyl-CoA that's accumulated through beta oxidation, you generate ketones. You go through ketogenesis, so you convert that acetyl-CoA to ketone bodies through ketogenesis. Those ketone bodies, like I mentioned in the previous slide, are this backup fuel source when glucose is low. Ketone body oxidation is then taking those ketone bodies and doing something with them, oxidizing them. So they can be broken down into carbon dioxide and acetone or oxidized back to acetyl-CoA. So I wanted to kind of help you visualize how these different processes relate together. So we're starting with lipids, specifically the monomer of lipids, triglycerides. We can break those down into individual fatty acids and glycerol through lipolysis, the lysing of lipids. Beta oxidation takes those and turns them into acetyl-CoA. So we have this acetyl-CoA as a central intermediate. That can then be used to form ketone bodies through ketogenesis. When you have the oxidation of those, ketone body oxidation, you can either produce carbon dioxide and acetone or continue on to acetyl-CoA, which can then be transitioned back into fatty acids and glycerol through lipogenesis. And you have kind of this full coming back of all that process. So when we're thinking about diabetic ketoacidosis, I wanted to explain a little bit about that condition. You might have heard of it before, depending on what types of patients you've worked with. But ketoacidosis, when we break apart that word, keto is ketone, acid is acidic or low pH, and then an osis is some kind of problem, so a general bad condition. Some of the symptoms of this include nausea and vomiting, fatigue, drowsiness, thirst, weight loss, abdominal pain, hunger, and increased urination. So some of these things are just kind of classical diabetes symptoms, but if you have a patient who's not monitoring their insulin, these are some symptoms to look out for. So what's happening here is when you have too much ketosis, you are having a lot of this accumulation of ketone bodies, and that causes this very low blood pH. Remember, there's this relationship with ketone bodies and carbon dioxide, and when you have too much ketone bodies, you have a very decreased blood pH, which is a problem. So remember, whenever we're thinking about a condition, some pathology, we're thinking about what is being thrown out of balance. And usually, if you have homeostatic balance with insulin and glucagon, insulin is functioning to do a lot of things, but also it inhibits lipolysis. So you don't have that breakdown of those triglycerides into their constituent pieces. So remember, the products of lipolysis include, so you're breaking apart triglycerides into monoglyceride or glycerol and fatty acids. A lot of things can happen to those fatty acids afterwards. So a lot of processing, a lot of acetyl-CoA and ketone body accumulation. And if we think about this formation of ketone bodies and subsequent ketoacidosis of too much formation, we know that that makes an appearance in people with type 1 diabetes. So we want to kind of think about why there's a risk there, what is being thrown out of balance. And so remember, insulin usually inhibits lipolysis, but then when you have type 1 diabetes, you're not producing enough insulin. Either you're not producing it at all due to pancreatic damage, or you're not producing enough of it. So if you don't have insulin inhibiting and providing negative feedback about lipolysis, then you're going to have increased rates of lipolysis. That means that your triglycerides are going to be broken down into fatty acids and monoglycerides. That accumulation of those tiny pieces means that there's going to be higher rates of them being oxidized to acetyl-CoA. So with beta oxidation, we get too much of that. And so then we're going to be having these really high levels of acetyl-CoA that has to be processed further. So we have these high rates of ketogenesis, the formation of ketone bodies. So when we have all these ketone bodies that are accumulating, that's going to decrease pH and cause ketoacidosis. So all of this comes down to the fact that people who are diabetic don't have that insulin to provide negative feedback. So at the end of the semester, when you're working on your term paper, it's going to be really important to identify what is throwing things off. What's the mechanism that actually causes all of these other symptoms? Because of course, having nausea and vomiting and weight loss and increased urination are going to continue to create problems, but there's a single mechanism, that lack of insulin, and this whole kind of relationship with lipids and acetyl-CoA that is really the crux of that mechanism. This is kind of a summary of what I just said. Insulin inhibits lipolysis, but people who have type 1 diabetes, so they're insulin dependent, they have to take insulin, also called diabetes mellitus or juvenile onset. So type 1 diabetics can't produce insulin or don't produce enough due to a problem with the pancreas. And oftentimes people monitor their insulin. There's a lot easier ways to do that now with, those those monitors that you can wear and that automatically provide you with insulin. But it is very challenging for people to constantly have to check their blood glucose and then administer insulin shots. Especially if you're a child or someone who is dealing with a lot of other things, it's very challenging. And so if you don't manage your insulin, there's higher rates of lipolysis. That breaks down triglycerides into free fatty acids and monoglycerides, which are then oxidized to acetyl-CoA through beta oxidation. This excess buildup of acetyl-CoA accumulates. When you have excess of things, it gets packaged away, so it's converted to ketone bodies through ketogenesis. You have these accumulation of ketone bodies, that lowers the pH of blood and causes ketoacidosis. So ketone-based acidosis, a low pH problem of the blood. So this is kind of an image that's summarizing a lot of the different things that we've talked about today and kind of what happens when you throw things off, specifically in the case of diabetic ketoacidosis. So this might help you visualize this. You don't need to memorize this whole diagram. You wouldn't have to label it on an exam, but it is helpful for understanding the case study. So I hope that this lecture made sense. If you have any questions, you are always welcome to email me or touch base with me in class.